I would like to introduce our webinar speaker, Dr. Kirsten Heiss. Kirsten received her PhD in infectious diseases from the Ruprecht-Karl University in Heidelberg in 2006, working on the malaria-causing agent Plasmodium. Further postdoc studies at the Institute of Immunology at the University Hospital of Hamburg-Eppendorf enabled her to combine infections with immunology. Back in Heidelberg in 2009, working as a scientist at the Center for Infectious Diseases at the University Hospital of Heidelberg, her research interest focused on vaccine development and the identification of novel targets for intervention strategies against malaria. In 2018, Kirsten joined the R&D unit of Pepperprint and is responsible for the discovery of novel antibody-mediated biomarkers in the field of autoimmune and infectious diseases. So the title of Kirsten's presentation is Autoantibody Signatures in SARS CoV-2 infected and vaccinated individuals. So once again, please use the questions pane and type your questions if they come to mind. And we will also share the slides and the post-webinar file. Kirsten, I'm handing over the cursor to you. Please take it away.
Thank you, Kirsten, and welcome everybody also from my side to today's webinar addressing the topic of the association of SARS-CoV-2 infection and autoimmunity. So in this context, I will present our pilot studies that we performed on the screening for autoantibodies and the analysis of antibody responses to epitopes that are shared between SARS-CoV-2 and human proteins. So starting with a brief introduction. So COVID-19, as we all know, is a respiratory infection. However, it can result in complications affecting multiple organs. So immune responses can both protect against the disease as well as drive it. And I guess here the termini of the double-edged sword which fits quite well. So here on the left-hand side in the figure from Rosella, Tarlotta, and co-workers, this nicely illustrates the immunological pathways that may be activated by a SARS-CoV-2 infection and accordingly how SARS-CoV-2 may trigger autoinflammation and autoimmunity. So there are different immune-mediated disorders have been already described in the literature in SARS-CoV-2 infected subjects, for example, myositis, myocarditis, and also vasculitis, and there are even more. And several studies reported the circulation of autoantibodies in COVID-19 patients. While in general, viral infections are known to be associated with or even worse than autoimmune diseases, and possible mechanisms proposed for viral-induced immunity are molecular mimicry and bystander activation, which are nicely illustrated here in this figure. So in the situation A, we have the normal situation where immune responses against the infectious target are induced, leading to viral clearance. And in B, for molecular mimicry, we have antibody or immune responses towards epitopes that are shared between the virus and human proteins, leading ultimately to cross-reactive responses, so leading to targeting of self-antigens. Last but not least, bystander activation. Here, autoreactive immune cells are going to be activated due to the release of self-antigens, which are under normal circumstances not seen by the immune system. So I want to highlight in this figure the molecular mimicry part, since we measured antibody responses towards such shared epitopes. Well, in the previous slide, I already pointed to the presence of autoantibodies in COVID-19 patients, and I would like to add a few more words to that. So here on the left-hand side, you see a table where some of the autoantibodies that are have been found in the association with COVID-19 are listed together with the autoimmune disease or disease syndrome secondary to a COVID-19 infection. Of course, the list is not complete, so there are obviously more autoantibody reactivities present, as for example, it was also observed autoantibody reactivities against cytokines, as for example, anti-interferon antibodies, and also autoantibodies targeting host cell receptors. Here, as an example, autoantibodies against the SARS CoV-2 host cell receptor ACE2. So for some of these autoantibodies, there is a possible association with life-threatening SARS-CoV-2 infection. So for example, it's discussed in the literature that the presence of anti-nuclear autoantibody might be associated with a poor prognosis for these patients. So for others, however, the clinical significance is still unclear. And it is tempting to speculate that potentially there are more autoantibody reactivities existing which are not yet described. And in the context of screening for novel autoantibody reactivities or for screening of antibody reactivities in general, I would like to introduce to you peptide microarrays as a screening platform, allowing the screening of patient sera against large libraries of peptides. And this is illustrated in this figure. So we have the proteins of interest that are translated into overlapping peptides and printed onto glass slides, which then can be screened against sera or also purified antibodies, depending on the project, and later analyzed to identify peptides of interest. So you will get antibody profile, and of course you can measure different antibody isotypes, either single or also in dual readouts. So now how this printing exactly works is shortly highlighted here in this slide. So we at PeppaPrint, we have a laser printer that has 24 printing units, one for each amino acid, which prints peptides directly onto glass slides through a series of melting and coupling cycles. So, and finally, we can produce peptide microarray, either displaying linear peptides or conformational peptides. But now coming to our study, which is divided in two parts. So in the first part, we screened COVID-19 patient sera and healthy control sera against human autoimmune disease associated linear epitopes. So we used a peptide microarray that covers above 3,000 human autoimmune disease associated B-cell epitopes, including peptide variants with post-translational modifications, which are known or described in the context of autoimmunity. So this B-cell, peptide library contains peptide variants with citrulline as well as acetylysin modifications. Moreover, this peptide collection, of course, covers a variety of autoimmune diseases, as for example, so includes B-cell epitopes that are associated, for example, with multiple sclerosis, diabetes, rheumatoid arthritis, SLE, etc. So the list is even longer. So now what we now use to screen that peptide microarrays, these are our patient cohort that we used. So we had, of course, a control cohort with SARS-CoV-2 negative patients or individuals, and we have two different COVID-19 patient cohorts. One with patients with mild disease progression versus a patient cohort with severe COVID-19 disease progression, meaning here all patients with acute respiratory distress syndrome. And here we profiled IgG and IgA in parallel. In the second approach, we were interested in antibody responses against epitopes that are shared between SARS-CoV-2 proteins and human proteins, and this in two different scenarios. So first, in the context of infection, and in the second hand, in the context of COVID-19 vaccination. So in the context of infection, what we did, we first tested our cohort for a general immune response against the proteome of SARS-CoV-2. And for that, we used a peptide microarray covering the entire proteome of SARS-CoV-2 as overlapping peptides. And we screened for IgG and IgA profiles. And then in the next step, we designed an array that covers 90 SARS-CoV-2 peptides that were described in the literature with homologous sequences in human proteins. And these 90 peptides were spanning across the entire protein, so originating from different SARS-CoV-2 proteins. In the context of vaccination, we tested pre and post-vaccination sera, and for the post-vaccination sera, we got access to vaccination sera after the second shot. And here we were first interested to know the vaccine-induced antibody response against spike on epitope level. And for that, we used a peptide microarray that covers the spike protein as overlapping peptides from all coronaviruses. And in the next step, we also tested antibody responses against the shared epitope with using the same array as I explained for the infected cohort. But here later on in the analysis, then we focused on the epitopes derived from the vaccine candidate spike. But now coming to our results, and this is just as a reminder. So first, we analyzed the autoantibody signature in COVID-19 patients. Just to remind you, here we use this array covering above 3,000 autoimmune disease-associated epitopes. And you may pay your attention here on this representative staining images from the different disease cohorts we analyzed. So what we here see in red are IgG responses, and in green IgA responses, and the arrays are framed by control spots. And what you can see, and I hope you agree with me when you just look at these images, is that you have a stronger response in the severely ill patients. And this is indeed what we see when we quantify the data for all patients. And this is shown here in a heat map where you have all the peptides of this array on the horizontal axis, whereas you have all individuals from the different cohorts on the vertical axis. Above, you see the results for IgG, below for IgA, and the color code indicate with the black color always are low intensities, going up to the colored one, either in red for the high intensity values for IgG, or in green, the high intensity values for IgA. Overall, what we observed is was a heterogeneous epitope recognition pattern between the study groups, with a far stronger response in the severely ill patients, and especially in the case for IgA. We were next interested, and we next determined the most differentially recognized epitopes, and I will present you our results for epitopes that show a strong reactivity in severe COVID-19 patients compared to mild COVID-19 and healthy controls. So this is our list of these differentially recognized peptides in severe COVID-19. And what you can also see is the respective assignment to the antigen. And we have here again the reactivity pattern of the individuals from the different cohorts. The color coding is the same, going from intensities, so low intensities in black, going to the colored one to the stronger signal intensities. Overall, what we detected are numerous epitopes derived from nuclear autoantigens, as for example, a ribonucleoprotein or La protein, or also the topoisomerase. Furthermore, there are epitopes which we can classify as tissue associated. So for example, from the thyroid gland or from the central nervous system. And we have a minor group with a few peptides, which we can group as immune system associated, as for example, this interferon alpha-2, T-cell receptor, etc. So overall, we found several autoantibody reactivity described for autoimmune diseases like SLE, vasculitis, and Graves' disease. So autoimmune syndromes that were found to be associated with COVID-19. I'm now switching to the second topic on antibody responses against the shared epitope between SARS-CoV-2 and human proteins. And we are going to start with the study on infected individuals. And here we were first interested to know the antibody response across the entire proteome of SARS-CoV-2 elicited after infection in our study cohort. For that, we used the array covering the entire proteome of SARS-CoV-2 as overlapping peptides. And here you can also see a representative staining images of SARS-CoV-2 naive and SARS-CoV-2 infected individuals. Again, the red dots indicate IgG responses, whereas green indicate IgA responses. And whenever you see, of course, an overlay or yellow, it's an overlay by both isotypes. And again, the arrays are framed by control spots. So after quantification, the data are looking like that. So here the data are displayed in an intensity plot showing representative IgG response patterns for 4 infected individuals, so the colored lines across the SARS-CoV-2 proteome, and the median here of our healthy control is the dark gray line here below. So what you see on the x-axis are the overlapping peptides of the SARS-CoV-2 proteome, and here with the assignment to the respective SARS-CoV-2 protein. On the y-axis, you have the IgG fluorescence intensity. So we detected for all individuals that we analyzed, we detected a proteome-wide antibody response. So it was spread across the entire proteome. And as we and others have shown in previous studies, the breadth of antibody responses vary among individual subjects. So now we went on to analyze the antibody responses against the shared epitopes, and this is shown here. So again, you have a representative staining image showing above some images from the infected versus the control cohort. And here you can see a different array format, having multiple array copies on one slide. This always depends on the number of peptides that are printed on the respective array. So the further data analysis revealed the following picture, which are again shown in a heat map where you have again, the list of peptides that are covered by the array and the respective SARS-CoV-2 protein where this peptide belonged to. And here the reactivity pattern again of the different individuals from the different cohorts. So in summary, what we observe here is that in infected individuals, we observed a diverse epitope recognition pattern in our cohort with epitopes that are not recognized at all. For example, here in this upper part, we have epitopes recognized by only few infected subjects, like here. And also we have a handful of epitopes that are recognized more frequently. And to have a closer look on the latter ones, we selected now for those epitopes recognized most frequently in infected individuals, which brought us to this picture here. So it's the same representation. So again, you have the list of peptides, the SARS-CoV-2 protein, the reactivity pattern of the individuals from the different cohorts, and this time also the human protein with the homologous sequence. What may catch your eye are responses against these two epitopes here, with homologous sequences in olfactory receptors, having in mind anosmia as a well-described disease symptom in COVID-19. And I would like to point out another frequently recognized epitope, and this is this one here, where it has been shown recently in a publication that this epitope can bind to various HLAs, including those associated with autoimmune diseases. So in the last couple of minutes, I'm going to present our data on the vaccination-induced antibody response, and accordingly also the antibody response against the shared epitopes. So we first were interested to know the vaccination-induced immune response against spike, so the vaccine candidate. And for that, as I mentioned in the beginning, we used the array covering the spike sequence as overlapping peptides from all coronaviruses. And what you can nicely see here are two examples from two vaccinees, pre and post vaccination. Again, you have in red IgG responses and in green IgA responses, and here the control frame. And what you can nicely see, and I hope you agree with me when you focus first on this upper part where the sequence of SARS-CoV-2 spike is located on the array, you see a nice induction of epitope-specific IgG responses after vaccination. And what you can also see, and this is what this chip offers, you can also see whether the vaccination induced cross-reactive antibody responses to other coronaviruses. And here you can see, for example, one of those, and this is due to the high sequence similarity between these two coronaviruses in this particular region. Now we went on and we also quantified this data, and I am now showing you the results for 3 vaccinees just to have the entire picture. And this is again shown in an intensity plot where you have the spike overlapping peptides from the different coronaviruses on the x-axis and the IgG fluorescence intensities on the y-axis. And here we have the 3 vaccinees. The brownish line always indicates the responses pre-vaccination, whereas the black dotted line always represents the antibody responses post-vaccination. And here you can see this for SARS-CoV-2 spike. What you also have already seen in the staining images, you have a nice induction of epitope-specific IgG responses. And what we also have seen already in the images, you have here this induction also here, or these cross-reactive antibodies to sequences from SARS-CoV spike. Finally, from the last part, I'm moving now forward to the analysis of antibody responses against the shared epitopes again, with the focus on spike, since of course all individuals were vaccinated with spike. And here you can also see representative staining images, and I guess you can already see it from the image. There is not much happening, which is good. And this is also the case when we quantify the data, which you can see here on this heat map. So here you have all the spike epitopes that we selected. And you see that for most of the shared epitope, we detected no antibody responses at all. There are only a very few number popping up, but here in this case, for example, you already have these responses pre-vaccination. And we only have here against this epitope a weak antibody response post-vaccination. So if we now compare this cross-reactivity patterns between infected and vaccinated subjects for spike, it looks like that. And I guess it's clearly visible that you have more frequent cross-reactivity in infected than in vaccinated individuals. So this already brings me to the conclusion. So going back to the first part where we screened for autoantibody signatures, here we observed in our cohort a heterogeneous epitope recognition pattern between the study groups with a far stronger response in severely ill patients. For the severe COVID-19 patients, we identified numerous IgG and IgA-specific autoantibody reactivities. And we had several of these epitopes that can be assigned to nuclear autoantigens, others made to more as these tissue-associated autoantigens. Of course, studies with larger cohorts are needed and also from different time points, so longitudinal studies to further confirm the identified epitopes and also to get an idea of the clinical relevance of these epitopes. From the second part, where we analyzed the antibody responses to these shared epitopes between SARS-CoV-2 and human proteins. So here in the infected individuals, we observed few antibody responses to the selected peptides shared between SARS-CoV-2 and human proteins, whereas in the vaccinated cohort, there were no antibody responses against most of the selected peptides seen. When we compare infection versus vaccination, we have a more frequent cross-reactivity after infection compared to vaccination. And last but not least, whether or not these antibodies recognizing either of the epitopes I here have chosen this DDD epitope have a clinical relevance, of course, remains to be determined in future studies. And with that, I would like to thank you for your attention and for joining the webinar, and we are happy to answer any questions.
Great, thank you very much, Kirsten. So once again, to the audience, if you do have any questions, please use the questions box and type them. And there were already some questions that we received. I'm trying to read them in the order as they appear. So the first question, bear with me. Here someone asks, Kirsten, there were some signal in naive samples in the upper left corner. Are these sticky peptides or shared or low complexity epitopes from other antiviral antibodies?
You mean now from the, this is not a question from which approach, right?
I think that question came in a little bit in the beginning, you know, so I'm not 100% sure to what exactly refers to your presentation.
It would be interesting whether it refers to the autoimmune chip, because here, of course, for autoantibodies and also in healthy individuals, it's known that also healthy individuals have autoantibodies in the serum present, although they don't have an autoimmune disease. So here, the background in the healthy control is something that we would expect. Yeah, if it goes to the background from the SARS-CoV-2 proteome chip, yeah, we also see here some cross-reactivities in naive individuals, and what we always see is this stretch in the NSP3 region, which always pops up in the naive control, for example. But whether or not these are now very sticky peptides, I honestly have to go back to the structure of the epitopes and the sequence.
Yeah, maybe the person who asked the question can specify this a little bit more. And again, we'll be sharing those slides after the presentation. So here's another question. Which vaccines are used for this data? Can you disclose this or say anything about this?
So So we included mRNA vaccinase and also vector-based. So they are all in.
Okay, great. So here's another similar question. Do you also look at specificity for anti-PF4 antibodies, which are reported to be increased in TTS cases after ChAdOx1 and COVID-19?
I guess here I'm a bit lost, to be honest.
Yeah,
I guess here we need a bit more details what exactly the question refers to.
okay, so also going back to the person who asked that question, maybe you can specify this question in more detail. And again, if you don't get to it during this presentation, we will answer you offline. Okay, hold on. Scrolling down further here, questions still coming in. Someone is asking, where did you obtain the patient samples? Was this a collaboration with the clinic?
No, this was no collaboration. I guess you refer now to the COVID-19 infectious samples.
Most likely, yes. I'm also guessing.
No, here we bought the samples. So they were from serum suppliers.
Okay. Here's another question. Do you have a negative control for the autoantibody induction? Could it be a very general induction of self-antibodies?
This is now how you would define really a negative control, right? So I guess in the autoimmune field, what would be the best negative control? Good question, very good question. But no, we don't have.
That's my understanding, I guess.
No, we don't have such a control.
Mm-hmm. Can you maybe comment on the controls in general that are on the chip?
So on the chip, so we used healthy controls where no autoimmune disease, when it now goes to autoimmunity, when no autoimmune disease was diagnosed, and also, of course, that were SARS CoV-2 negative. So they were collected before the pandemic.
Okay. So here's another question. Any data on persistent COVID-19 autoimmune response, in parentheses here, long COVID and also PASC, not sure what that means, and or breakthrough? Okay, yeah. So maybe the long COVID part. So again, any data on persistent COVID-19 autoimmune response, referring as long COVID, and or breakthrough post-vax infections.
Well, yeah.
Also, yeah. Hold on, there's another part to it.
Oh my goodness.
Hold on, let me just. Also, do you think IgM autoantibodies are important to pursue?
Okay, first of all, the question of long COVID, I totally agree. It would be very interesting to look at the reactivity over time, because of course we, in our little pilot study, we only looked at one time point, right? And it would be, of course, very interesting to know whether these reactivities are stable over time. But we haven't done that, so, and we don't have yet the samples for that, but it would be very interesting to do such a longitudinal study. And also for the middle question, with respect to breakthrough infection, also very interesting to go in detail into that, because we are faced with it in the moment that we have breakthrough infections. But so far we don't have hand-ons on the samples, but it would be very interesting to study this samples. Yeah.
Okay.
And the last question, it was IgM.
Right. And there was some.
Yeah, sure. Well, good question. So in autoimmunity, of course, different isotypes are under investigations, right? And we now focused on IgG and IgA, but of course, would be also great to have a look on IgM as well. Yeah, sure.
Great. So here's another question. How was the population selection controlled for pre-existing conditions? This is with regards to autoantibody analysis.
Good point. This was not yet controlled in this. No, it was not controlled. So as we bought this samples, and of course we cannot. No, we don't have any information about that.
Okay. Here's another question.
Yeah.
Have you done these screens also with cyclic peptides? And if so, are the responses different?
No, not yet, but also quite interesting to do so. Yeah, but we haven't done it yet.
Okay. Yeah. Then someone asks here, any differences between the two types of vaccines?
In case of the induction of epitope-specific responses, no. There is no difference in epitope pattern. So you cannot really say, okay, this vaccine induced that and that induced that. But I also have to mention, of course, that our study cohort with 6 vaccinés was not that large. So of course we need a larger cohort to really go into detail into that.
Great. So I can say the interest is very nice from the audience. Thank you very much for asking all the questions. I'm afraid we won't get to all of them. Here's another question about normalizing samples. So do you have to normalize patient plasmas and healthy controls? And how is this being done, just roughly?
To normalize, so usually when we do the data analysis, we for example for significant peptides, we of course do normalization and then statistical analysis.
Okay. Here another question about the vaccines. Do you see a difference in autoantibody profiles after mRNA vaccine and vector vaccine?
No.
Okay. So let's do 2 more questions. Here's a short question. Is it better to use purified IgG rather than plasma or serum?
Good question. Well, I haven't done this whole entire analysis, not with purified IgG, but I guess the plasma works quite well. So.
Okay. So I'm going to read the last question. We have to try and wrap this up slowly here. So COVID-19 patients have some vasculopathies, as do patients with IgA nephropathy. Is the high IgA response of COVID-19 contributing?
Well, I cannot comment on this. I guess we cannot conclude that from our studies. And this is more a mechanistic question, right? So.
Right. That's what how I would understand it.
Yeah.
Great. So once again, let's begin to wrap this up. So all the questions that remain unanswered, we will try and comment offline. So once again, thank you all for attending this webinar presentation, and we also want to thank our speaker. Once again, we will make the presentation slides and webinar file available. If you have any questions, please contact the PepperPrint team directly. Please also check our website for updates, future events, and webinar announcements. So I will end this webinar now. Thanks again all for dialing in.